Good morning, students.
Sir.
Okay, so we shall wait for 2 more minutes. Only four of you are there, right? Okay, we wait for 2 minutes. Should we start, students? Students, should we begin? Students,
Yes, sir.
do you think someone else will join? No. Okay. So the topic that we were supposed to learn today was your second category of the mutagen, that is chemical mutagens, right? So this is B types of mutagen. One was the physical mutagen, where we have seen the role of ionizing radiation and the non-ionizing radiation. The second category will be your chemical mutagens. Now, as the name suggests, these are certain chemicals that has the potentiality to impart mutation in the DNA. Okay. Point number one, you write chemical mutagens are chemical mutagens can be classified into can be uh, can be uh, clubbed into, or you write down, can be classified into three categories based on their action, right? First category is your, we'll write, first category is your some chemical mutagen acts as base analog and replaces a right base. Okay, some of the chemicals mimics the ATGCs and they take the position of those ATGCs and subsequently they causes the mutation. The second way of creating mutation is directly reacting with the directly reacting with the nitrogenous bases of DNA. So they generally causing structural changes. So they directly replaces the nitrogenous bases that causes structural change. And the third mode of action of this chemical mutagen is that they act indirectly by promoting other chemicals to cause mutation, right? So they are not acting directly, but they are acting indirectly. Okay, now let's understand how does the base analogs react. Okay, so we'll see how base analogs actually causes mutation. Now, as the name suggests, base analog is a category of chemical that mimics the structure of nitrogenous bases, and somehow or the other, instead of the right kind of the nitrogenous bases, these modified nitrogenous bases are included into the DNA strand, and subsequently their pairing nature changes and they impart different kind of bases. So in the subsequent daughter cells, the mutation is imparted, right? So I'll share a diagram in the WhatsApp group. All of you draw that diagram. Then we'll see how the base analog reacts. Right? We'll try to see a classical example of base analog that is known as 5-bromouracil. Okay. Now the diagram. Can you see the diagram in the WhatsApp group? Can you see the diagram in the WhatsApp group? Yes or no?
Yes, sir.
So draw the diagram. We'll discuss the example of 5-bromouracil. Okay, I am giving you 2 minutes. Have you finished, student? Students, have you finished? Students, have you finished the diagram? Can you respond, one of you?
Wait, sir.
Okay. Just draw the diagram.
Done, sir.
Okay, good. Now see, the case that we are studying is of a base analog. Now as the name suggests, the base analog is a chemical that has got structural resemblance with one of the nitrogenous bases, that is A, T, G or C. In this particular case, we are discussing a base analog that is known as 5-bromo-uracil. Okay, in short it is known as 5-bromo-uracil. Right, now this is a chemical compound which is a potential mutagen or chemical mutagen. This 5-bromo-uracil falls under the base analog category, and by virtue of being similar in structure to thiamine, they causes mutation. Now understand what happens. If you can focus into the diagram, they have shown an example of A and T in two respective strands of the DNA. Once the replication is done, as the 5-bromo-uracil has a structural similarity with thiamine, instead of a thiamine in the cell, a 5-bromo-uracil is incorporated. Okay, in the second diagram, the bromo-uracil has been added, right? Now what happens, bromo-uracil, if it is available in the normal wild form, that is the keto form, then nothing will happen, no mutation will happen. But if the rare form of the 5-bromo-uracil, that is the enol form, if the bromo-uracil is now converted into an enol form because of a tautomeric shift, the star has been given. Instead of the initially bromo-uracil has been shown in the diagram as a Bu, the third picture, the third portion of the diagram has a star in the bromo-uracil. That means there is a tautomeric shift and the bromo-uracil available in the strand is now shifted to a rare enol form. If that happens, then in the subsequent replication, two daughter strand will be formed. In the first daughter strand, A was binding with T, no mutation. In the second daughter cell, daughter DNA, you can see that bromo-uracil, although it has replaced the thiamine, so by virtue of being a thiamine replacement, it should have bound with a adenine, but it has a affinity towards a guanine. So instead of a adenine, a guanine has come. So this particular mutation is inherited, and in all the subsequent replication, this guanine and 5-bromo-uracil, that means 50% of the daughter strands will be mutated. Am I clear? Am I clear? If you can see the diagram, it will be comfortably you can understand. So you write down two important statements about 5-bromo-uracil. Write down 5-bromo-uracil looks like thiamine and replaces a thiamine in the DNA strand. Point number 2, if the wild form of 5-bromo-uracil, that is the keto form, is present, no effect is seen in the subsequent daughter DNAs. Okay. However, if the 5-bromo-uracil is converted into 5-bromo-uracil star, that means rare enol form, because of a tautomeric shift, it creates mutation in the subsequent generations, right? Why it happens? It happens because the 5-bromouracil star, that means the enol form, pairs with guanine instead of adenine, because it was replacing thiamine, so it should have been bound to adenine, but now it binds to guanine, and in the 50% of the subsequent generation, it imparts mutation because a guanine will replace a adenine. Okay, this was example number 1 of base analog type of mutagen. Example 2 will be your 2-aminopurine. Just understand the name, just remember the name, 2-aminopurine. We will not discuss the mode of action. Example 1 was 5-bromouracil, example 2 is your 2-aminopurine. Okay, so this was the first category of chemical mutagen. Now we'll see 3 more types of chemical mutagen. First one is your base analog, right? Base analog. The second category is your deaminating agents. They also act as chemical mutagens. So the characteristics of deaminating agents is it creates point mutation. It creates point mutation by removing an amino group. As the name suggests, it is a deaminating, so it removes the amino group from the bases and thus imparts a point mutation. Example of such kind of deaminating agents are nitrous acid, just understand till this point. Example is nitrous acid. Okay, the third category of your chemical mutagens are alkylating agents. Okay, they impart mutation by adding a alkyl group to the nucleotides. Okay, example will be your ethyl methane sulfonate, EMS, or ethyl methyl sulfonate. Most of these chemicals you will use in laboratories. Okay, then nitrogen mustard and dimethyl nitrosamine. These are example of alkylating type of chemical mutagens. Dimethyl nitrosamine. Okay, so these are the three examples of alkylating type of chemical mutagen. Okay, then the last category of chemical mutagens are intercalating agents. What are intercalating agents? These agents are so small in structure that they can intercalate. Intercalate means slip. They can slip through the nitrogenous bases in the DNA. So you remember that 3.4 Armstrong length of the 10 nucleotides. So these chemicals actually are so small that they can intercalate through the nitrogenous bases. And a very classical example is your Etherium Bromide. We use Etherium Bromide in our gel casting, right? You remember that orange color chemical, which we say that it is a highly carcinogenic or a mutagenic substance. And they causes mutation to the DNA because they can slip through the nitrogenous bases and they can dislocate the pairing. Okay. Write down intercalating agents. Intercalating agents creates single nucleotide insertion or deletion. Example, there are numerous examples that you use frequently in the laboratories are proflavin, then comes your acridine, these are dyes, acridine, then acridine orange, these all are dyes, then your Etherium Bromide used in electrophoresis, gel electrophoresis. Okay, so this was all about the chemical mutagens. In next class, we'll see the biological mutagens. We'll be more focused in the biological mutagens because time and again you will find numerous viruses that causes mutation or cancer by integrating their genome with the genome of the human being. So we'll see how viruses can also cause mutation. Okay, so we'll stop the class at this point, right? So the third category will be C, that we'll discussing tomorrow. Biological mutagens. Okay, students?
Okay, sir.
Okay, so we'll stop at this point. I believe that you have written the examples and types of chemical mutagens, right?
Yes.
Okay.
Yes.